Hi, welcome to Cellular Reproduction. This week we'll be going over Chapter 10 of the book. So first let's talk about the genome in prokaryotes. So in both prokaryotes and eukaryotes, we have a genome, which is just packaged DNA. Within prokaryotes, the genome is found within a loop or circle within a nucleoid because they don't have a true nucleus. They also have smaller loops called plasmids, which are not essential for growth, but they can be exchanged with other bacteria or organisms to add to their chromosomal DNA, which leads to things like antibiotic resistance and that adaptation being able to be passed on to other species. In eukaryotes, we have two different types of cells. We have somatic cells, meaning body cells, which have 46 chromosomes, and we have our sex cells or gametes with 23 chromosomes. Our somatic cells have two homologous sets of chromosomes, one from each parent, and those are known as diploid cells because they have two homologous sets. Our gametes are haploid because they only contain one set of chromosomes. So sometimes diploid is abbreviated 2N and haploid is abbreviated 1N. So they are called homologous chromosomes because they are the same length, and these genes sit in the same locus. So a gene is a functional unit of chromosome, a protein, and a locus is a location along that chromosome. So those functional units sit in the same place in homologous chromosomes. Genes are going to determine characteristics of an individual. They won't be identical, since you do have one set from each parent. And traits or alleles are going to be variations of those characteristics. Variations of traits in individuals leads to genetic diversity, which is great because, as we will talk about later, genetic diversity is extremely important for preventing mutations from occurring, allowing our species to adapt and thrive. The DNA has to be pretty tightly packed to fit in a cell's nucleus. Nuclei are small. DNA, when unraveled, can be quite long. So it has to be condensed and tightly packed to fit within that nucleus. DNA becomes condensed so that it's readily available when needed. When DNA strands wrap around histones, it forms chromatin. This DNA is going to replicate during S phase of interphase. And interphase is the time period prior to mitosis. During interphase, in this S phase, there will be two linked sister chromatids connected by a centromere. So let's talk about the cell cycle. The cell cycle is where cell growth and division occurs, and it results in the formation of two identical daughter cells. So this week we'll be talking strictly about the duplication of somatic cells. Next week we'll be talking about the duplication of sex cells. So with somatic cells, we are taking one cell and forming two identical daughter cells, genetically identical. Interphase is the phase that the cell spends most of its life in, and this is where the cell grows and the DNA is replicated. Mitosis results in the formation of two separate nuclei that are genetically identical. And lastly, with cytokinesis, that's when cells separate, forming two individual cells. So we'll show each step of that. So like I said with interphase, this is where the cell spends most of its life. There are three different phases of interphase. In the G1 phase, or growth phase 1, or first gap, This is when the cell will accumulate building blocks for DNA and accumulate energy that will be needed to duplicate the DNA. In the S phase, this is where we're going to synthesize the DNA and form our sister chromatids. And in the G2 phase, the second gap phase or growth phase 2, this is where the cell will replenish the energy and synthesize proteins that will be needed for mitosis. So mitotic division can be split into 2 steps: karyokinesis, or the formation of new nuclei, and cytokinesis, or the formation of new cells. There are 4 steps to karyokinesis: prophase, metaphase, anaphase, and telophase, often abbreviated PMAT. And in the book, they separate this into one more additional step, prometaphase, which is just an intermediate phase between prophase and metaphase. It's not always recognized as its own phase, so for the purposes of simplifying things, I'm not going to recognize it as a phase for the purpose of the lecture. In mitosis, our first phase is prophase. This is where the nuclear envelope will dissociate, chromosomes will move to opposite poles, and the sister chromatids will coil tightly together. The cells will start to elongate as we move into metaphase. Those chromosomes will align at the metaphase plate or equatorial plate like an equator. During anaphase, the sister chromatids will separate and pull toward opposite poles. And then in telophase, they will reach those poles and unravel. Nuclear envelopes will reform, and the result will be 2 identical nuclei. During cytokinesis, there will be a development of a cleavage furrow where the cells are still connected until the cells separate, which will result in 2 identical daughter cells. So cell division is not complete until the cells separate during cytokinesis. There is some difference with plant cells, as they have to form a whole new cell wall. So instead of a cleavage furrow, there will be a formation of a cell plate. In the G0 phase, this is also known as the quiescent or inactive stage, cells are not actively preparing to divide. This could be a temporary stage due to environmental conditions like not enough nutrients or the presence of certain growth factors. Cardiac cells and nerve cells are examples of cells that stay here permanently, which is why they have trouble repairing damaged tissue in the cardiovascular and nervous system, because they are not cells that divide actively. The cell cycle varies quite a bit, and it depends on the cell type, and the variation of time spent in each cycle will depend on the species and type of cell. Some cells have a shorter lifespan than others. The cell cycle can be regulated by external events, so the death of nearby cells may trigger the cell cycle. Not enough growth hormone or human growth hormone, HGH, may lead to the cell cycle being inhibited. Too much growth hormone may lead to the overactiveness of the cell cycle. The size of the cell, if the cell is too large, the cell will want to divide in order to be a little bit more efficient in its processes. And crowding of cells may also lead to an inhibition of the cell cycle. It can also be regulated by internal events. So, as we previously said, the daughter cells that are produced by mitosis are identical copies of the parent cell, the original somatic cell. So, there's a lot of regulation that takes place to ensure that those daughter cells are exact copies, because mistakes do happen. So, there are checkpoints throughout the cell cycle that prevent damaged cells from being replicated. If there's any mutations that occur, if there's any genes that are out of place, there are checkpoints to ensure that those cells will not go on to finish their replication. So there's different checkpoints. G1 and G2 checkpoints are within interphase, and they're to ensure that the cell is growing properly. G1 is also known as the restriction point. The M phase is near the end of metaphase, which is why it's called the M phase. And it ensures that the sister chromatids are attached to the spindle microtubules so that they're able to be pulled to either end of the poles during anaphase and telophase. If for some reason there are errors within any of these checkpoints, the cell will undergo apoptosis and will not continue to be formed. So, like we already talked about a little bit, the cell cycle can have positive or negative regulation. Positive regulation meaning that the cell cycle will be propagated, and negative regulation meaning that it will be inhibited. So there are different molecules that can propagate the cell cycle, different ones that can inhibit it. So cyclin-dependent kinases, or CDK, will propagate it. Negative regulation by the retinoblastoma protein, P53 and P21. Cancer is uncontrollable cell growth. So the cell cycle just doesn't stop. And cancer occurs due to mutations that accumulate and are unchecked during the checkpoints within the cell cycle. So there are what's called proto-oncogenes that code for positive cell cycle regulators. They can become oncogenes, meaning cancer genes. So these genes result in less functional proteins, which may prevent the cell from completing the cell cycle, which is great because we don't want malfunctioning proteins within our genes and being replicated. But these can also lead to cancers that occur. So CDK, the cyclin-dependent kinases, can be a proto-oncogene. Tumor suppression genes are segments of DNA that code for negative regulatory proteins. So they inhibit the cell cycle. They prevent this uncontrolled cell division. So, like we said with cancer, any mutations in the DNA could result in tumor growth and the formation of cancers. So retinoblastoma pigment, the retinoblastoma protein, P53 and P21 are all tumor suppression genes. So if they notice there is some kind of mutation that's occurring, they can suppress that from continuing to divide and form new cancer cells. In prokaryotes, they divide via binary fission. So karyokinesis is unnecessary. Remember, karyokinesis is mitosis, essentially, the formation of those nuclei. Prokaryotes don't have a true nucleus, so they don't undergo karyokinesis or a mitosis-like division. It's more so that one cell divides into two cells, kind of like budding, if you've seen that before. So binary fission tends to be less complicated and a more rapid process. It doesn't need histones or nucleosomes, none of the complexes that we had to make in order for mitosis to occur.